Hi, my name is Connor. I'm from the Li Lab.
This is how you make a page gel. There are different sizes of glass plates to make your page gel, but most of the time we just use the medium-sized plates, which are here. There are also different spacers, like the long ones here, or the short ones, and they have different thicknesses as well. So you can see some are thick and some are thin, depending on how much DNA or RNA you want to load into your gel. For the most part, we just use the thin spacers for the medium gel. Here is what you need to make one gel. So once you get your plates, you want to clean them with deionized water, just one side, and you can use a paper towel to wipe them like this. This can go in the garbage, and then you can do a better clean with the ethanol just to get anything else that's stuck to the gel. And then you can use a pin wipe with the ethanol. And that goes in the garbage. So while the ethanol is drying, you can set up your spacers. So the spacers go on either side of one plate of the gel plates, like this. Just make sure there's no crud on the spacers. They just need to dry for a little bit longer. Once they're dry, you can assemble the plates like this. Make sure you put the two clean sides together, and then secure it with the office clips. Okay, now we're going to make the urea page solution. So in this case, I will be making a 10% urea page solution, but you can make 5% or lower depending on the size of the oligonucleotide you want to load to the gel. So for a medium-sized gel with the thin spacers, I'm gonna add 50 milliliters of the solution. Next, you're gonna add 500 microliters of the APS, which is in this conical tube here. This should be in the mini fridge if it's not on the bench, and it should be in the top drawer here. And then you're going to add 50 microliters of the T-Med to initiate the polymerization of the acrylamide. Okay, once you add that, you kind of have to work quickly. So mix it for a little bit and then start pouring into the gel. I usually start at one corner slowly and then continuously tap on the plates to remove any bubbles that might form. You might get some drip, that's fine, as long as you put a paper towel underneath.
keep pouring until it reaches the end. Okay, sometimes you might get a little bubble like that. If that happens, you can grab one of these metal spatulas and just remove it. Okay, and then you can add your comb. Just make sure that there are no air bubbles. Don't insert the comb all the way in, just leave a little bit of space at the top so you can remove it more easily. So then you need to wait 4 minutes for it to polymerize, and then you can remove the comb and set up the apparatus. Okay, so once it's been 4 minutes, you can check the remaining solution in your beaker. If it's solidified, which it has, then your gel has probably solidified as well. So you can remove the clips and then take it over to the sink. Get a light dribble of water over the comb and slowly remove it. If you remove it too fast, then your wells might be warped or distorted. So make sure you do it slowly. Just rinse it off and then put it back with the others. And then use some deionized water to remove any gel bits that might be still on the gel. Okay, once that looks good, you're going to take it over to the apparatus here. So remove the covers like this, and then get your office clips, and you're going to make sure that the indented side of the gel plate is facing the reservoir at the top, so like this. And then there are two plastic things at the bottom, and you'll want it to just sit on it like this, and then line it up, and then clip it with the office clip. So you don't want the clips to be like up here, you don't want them to be down here, just level at the top is good. Okay, then you're going to add your buffer, which is 1 times TBE. There's TBE and TAE that we use in the lab, so make sure that it's TBE and it's 1 times. So you're going to fill the top reservoir until it covers the gel, and then you're going to fill the bottom reservoir till it just covers the bottom of the gel. And you're going to reassemble your apparatus here, and then when you're running the gel, it will produce a bit of heat, so you're going to want an aluminum plate to help disperse the heat, and you can just slide it at the back and clip it with the other office clips. So then you're going to plug in the leads to the power box. So red, positive into red, black, negative into black. Turn it on. The gel has to warm up for 20 minutes at least before you load any Legos into it. So you're going to set your wattage to 35, and that's kind of the maximum for one gel, or else it'll get too hot. And the voltage around 1,000 is fine, but it will max out at the wattage first usually. So then you're gonna turn it on, and if you have a timer, set it for 20 minutes, and then you can come back and load the gel. You also want to write your name on the gel or on a piece of paper towel, so if there's a problem with the gel, others can know whose gel it is. Okay, so once your gel has been running for 20 minutes, turn it off and remove the top lid here. So while your gel's been running, a lot of urea has accumulated in the wells, and before you load anything into them, you need to clear the urea out. So you can use the syringe like this, and you can just twist it off and pull up some of the TBE buffer like this. Screw it back on and remove it. Careful, the needle is very sharp. So just go into your wells and try and squirt out all of the urea. Like that. And you can just squirt the rest back into the reservoir like that. Okay, so when you're loading your sample, make sure that you have the correct volume that you want to load on your pipette, and make sure you're using the correct pipette tips. So for gel loading, we use these long tips to make sure that you can get into the wells. So I'm going to take some of my sample. It might take a few seconds for it to all get in the pipette tip because the end is very narrow. You're just going to make sure you go in between the two gel pieces and release all of the sample into the well. And then discard the pipette tip into the appropriate waste. And then once you're ready, once all your samples have been loaded, you can put the reservoir back on, the reservoir lid on, turn it on, and then start it again. Okay, once your gel is done, you can turn it off and remove the lids to the reservoirs and unplug them from the power box. So first thing you're going to do is remove the aluminum plate and put it back over here. And then to remove the gel, first you're going to put some paper towels down, and then just unclip one of the clips and open it up slightly just to let some of the buffer flow down, and then tilt it so some buffer will fall down with the gel. That way when you move the gel to the sink, you don't spill everywhere. So next you're going to remove the second clip, let it drain into the bottom reservoir, and then put it down on the paper towel. Then you can empty the buffer into the sink carefully. And just rinse the wells with tap water. Like this. And then that can just go on the shelf up here with the corresponding lids. Okay, now to get your gel out, you're first going to want to remove the spacers. So just push outwards with the spatula like this, and then pull them out like that. Do the same with the other side. Sometimes it can be hard to pull out. And then you're going to want to separate the gel from one of the plates. So just put your spatula between the two plates and pull up. And it'll either stick to the bottom one or stick to the top one, or in some cases it might stick to both. And in that case, you just have to be really careful when you're pulling it up. So you're going to want to get a piece of plastic wrap If there aren't any razor blades with the plastic wrap, there are some in this drawer here, and you can just open one. So pull some plastic wrap about this long and cut it almost all the way with the razor. Get a good hold of it and then rip the rest off. And slowly bring it over to your gel and cover it like that. Okay. Next, you're going to want to flip it over like this. And this is the hard part. You're going to want to separate the gel from the glass, the second glass plate. So the easiest way to do this is to find one of these corners, lift it up slightly, and then push this corner of the gel down, and then go along the edge and continue to push down like that until the bottom is separated. Then you're going to want to spread the plastic wrap with your fingers and slowly lift the gel plate up, and the gel should slowly separate from the plate like this. The lower the percentage of the gel, the harder it will be, so with a 5% gel, you're going to want to be extra careful. And then we're going to take our second piece of plastic wrap, cut it, and finish wrapping. Like so. Then your gel is completely wrapped and ready to be shadowed with the UV light. And then you'll also just want to clean up your workstation and wash your gels and put them back on the drying rack.